Okay, so for class 5, I've got the metalloregulation of bacteria and gram-positive bacteria. I'm talking about 5 different papers. A paper by a person by the name of Moore, looking at Bacillus subtilis soleus, gives a review on gram-positive response to copper stress. Rademacher talks about copper responsive gene regulation bacteria. So those 2 are reviews. Then there's 2 full articles, one by Reese Caballero. He talks about the ADCR protein in Streptococcus pneumonia. That is a zinc protein. And the last paper is by Aranda. Okay? So I'll just carry on. So the first paper, like I said, was by a person by the name of Moore, and it's the metal ion homeostasis in Bacillus subtilis, and this is just a review paper. So the first, and I'm just going to go through the figures from the paper. If there's a really important table, I'll try and pick that up too. Now, in this picture, what they're trying to show you is that Bacillus subtilis has both efflux as well as uptake proteins. On the left, you can see there's a number of uptake proteins. For example, the Mint proteins uptake manganese, and as for efflux, in Bacillus subtilis, you've got the copper Za complex. That efflux is copper. So there's a whole slew of input by Bacillus subtilis for metals. In this slide, they show that Bacillus subtilis has these proteins that also have homology to proteins in other organisms. For example, the iron uptake protein or ferrous uptake regulator is involved in iron homeostasis. It takes up iron in E. coli, Staph aureus, as well as Listeria monocytogenes. Per-R, known for taking up manganese and iron, is named for its peroxide resistance. Staph aureus, Listeria monocytogenes as well. And CJE would be Campylobacter jejuni, and zinc uptake, manganese uptake, and so on. So a number of them are already known. People who study the metalloregulatory proteins also want to know the selectivity of the metals. Sometimes a metal will activate the protein, but it's not always the same amount of activity. So the selectivity is also noted, and so they will make a list of descending enzyme activities. Okay, so Bacillus has a whole bunch of homologs to other bacteria. That's the point for this slide. In this slide, they're naming several of the uptake and efflux metalloregulatory proteins. For example, the ABC transporters MNT ABCD are very similar to the ones found in Streptococcus mutans slow ABCD. So they would be involved in the uptake of manganese. And just going through the operons, there's Mint, there's another Mint, COP for the copper uptake. Copper uptake is regulated by QR.
Uptake or efflux?
Sorry, efflux. Sometimes I say uptake for everything, right?
Yeah. You can say transport.
Transport would be easier to say.
Yeah.
There is known bacteria metalloregulatory proteins also for arsenic as well as for citrate. In this slide, they show the pathway for iron uptake and its storage. So there's a number of methods that the bacteria can input iron in the form of ferric citrate, ferrioxamine, ferrichrome, and so on. So these are methods that they...
This is also a
Is it-
review article then.
Correct.
Yeah.
Yeah, this is the current opinion of microbiology paper. So it's just a
Bacillus.
little bit of everything
In Bacillus.
in Bacillus, yeah.
Yeah.
So nothing really new other than this is everything we have.
Yeah.
A really common theme in metalloregulatory or transcriptional regulators is this idea that what you have are protein complexes that dimerize, whether it be an iron uptake or iron regulator rather, or zinc regulator. For the iron regulator, here's the motif. It's a I think it's a 11 base pair inverted repeat. Same thing with per R and zur R. That's a pretty common sort of theme throughout the
Among
metalloregulators.
Yeah.
And transcriptional factors too.
Yeah.
This is kind of an interesting little story in Bacillus subtilis. What they're doing is discussing the idea that the ribosomal protein L31, which is a subunit of the 30S ribosome, it's regulated by zur paralog. So normally RPME binds zinc, and when that's bound to zinc, it's going to bind the ribosome.
RPME is what? A transcription factor?
I believe so, yeah.
Okay.
In this case, L31 is transcribed, and YTIA is repressed by the zinc uptake regulator. So this protein is repressed. And the RPME would be the paralog of the zinc uptake regulator. So that's when zinc is being taken up by the RPME. If RPME loses its zinc, then it's not bound to the ribosome. What happens is zur is no longer repressing YTIA, and YTIA then takes the place of RPME, and RPME is then proteologically degraded.
What does R31 does, or why is it important to study this?
R31? The 30S ribosomal subunit.
L31?
I think so. Yeah.
Okay. It's okay.
We could take a look.
No.
I'll just pause what I'm doing. I don't know how to pause.
It's okay. Go on.
Okay. Carry on then.
Yeah.
So that's the first paper. The second paper talks about the response of gram-positive bacteria to copper stress. Now there's a number of known bacteria enzymes that have copper in them.
Yeah.
Cytochrome oxidase, dehydrogenase, plastocyanin, nitrate reductase, and so on, right?
Yeah.
I thought superoxide dismutase also contained copper.
Yeah.
But it's not on this list. I'm not too sure why.
Maybe by dismutase name? No? It does. In some bacteria, not in all.
Of course, yeah.
Like in strep mutans, the SOD doesn't require copper. It requires zinc as a cofactor.
I thought it would be on this table, though.
It should be.
Yeah. I would think that's a more famous example. All right, so here we have the Cupro enzymes and gram-positives. And as you can see, actinobacteria have a lot of Cupro enzymes. And they also have this MOCO, or molybdenum cofactor as well. So 38 genomes in actinobacteria at the time of this writing probably known, and 34 enzymes use copper and 31 use molybdenum.
34 of these actinobacteria have copper-containing enzymes.
Yeah.
They don't contain 34 enzymes. So out of the 38 genomes they have sequenced, 34 of them, they have copper-containing enzyme. It could be 1, 2, or, you know.
Right.
Yeah.
Yeah.
Why are they including this molybdenum cofactor?
Probably what they know. There's some problems with this.
Yeah.
Like, take a look at lactobacillus, streptococcus
Yeah, because
mutans.
they don't have any copper containing. So in Streptococcus mutans, they couldn't find any enzyme which requires copper as a cofactor for its activity. And it's not only the case of Streptococcus mutans, but most of the lactobacillus. So it's not...
That's not wrong to say that they're no users?
Yeah, they do not use copper for their metabolism.
I see.
Unknown.
This is interesting that they even included this one, molecules.
Just to show
no
that,
users,
yeah.
No, so there's nothing there,
no.
But they put it on the table anyway.
Just to show you that it's not necessary that all the bacteria requires cuproenzymes.
Right.
Copper is important for some bacteria, but not all, especially if they are facultative anaerobe or anaerobe. So in the beginning, when there was facultative anaerobe and aerobes. Do not one of them. I'm getting confused. Anyways.
Okay. A lot more is known about Enterococcus here than it is known about other bacteria gram-positives. But here's two models. So in the case of here, COP-A imports copper. COP-Z dimerizes with the presence of copper, and then it sits on the regulates the DNA for COP-Y. So it's a COP-Y.
COP-Y, usually in the absence of copper, it sits and negatively regulates the expression of this operon. When there is copper, COP-Z brings the copper. So COP-Y now binds to copper instead of zinc. So by binding to copper, it loses its affinity to bind to the DNA of the promoter and it detaches away.
Right.
And then this whole transcript is transcribed.
You can see also in the presence of zinc.
So usually it binds with zinc and then it sits on the promoter. But as soon as copper comes, zinc is removed, copper binds there, binds to COP-Y, and then COP-Y, the affinity of COP-Y binding to the promoter decreases and it detaches away from the promoter and the whole operon is. But they haven't actually shown in Enterococcus here that COP-A brings in copper. It's still a speculation.
It's a model.
Yeah.
Very similar model for Lactococcus, only instead of COP-A bringing in the copper, it's effluxing copper.
Both of them.
And in the case of Lactococcus, COP-B is regulated by this dimerized protein. Here's a list of the copper responsive regulators in gram-positives, and as you see, actinobacteria don't have this COP-Y type regulator, but they do have a CSO regulator. Same thing with bacilles and lactobacillus. Most of the regulators in bacillus is COP-Y.
Mm-hmm.
I came up with a structure for the COP-Z protein, and it has these two sides. It has beta sheets on one side.
Did you made this?
The alpha helices on the other side. And the bind together with this disulfide bridge.
Is it submutants?
No, I could show you. So you go into PubMed. No, not PubMed.
PDB.
Protein Data Bank, yeah. And here we are. This is the COP-Z.
COP-Z of what?
This is the copper chaperone of
okay. And
Here.
Okay.
Yeah, and you can just maneuver it, take a look at. So it's got those 4 beta sheets on one side and the alpha helices on the other side. And the serines of this molecule will dimerize. They'll form the disulfide bridge seen in this image there, using copper. So that was my picture. I kind of like the protein. Typically, the function or the structure of the protein in these transcription factors are a helix, turn, helix, wing. So they refer to it as a helix, turn, helix, wing. If I could follow it, it's kind of hard to see because there's 3 parts here. So if this is A1, there's A2, so there's 1 helix, 2nd helix, and here's the wing. This was an interesting story, I thought. I'd just mention that if you have copper bound to COP-Z, it will be in competition with other metals, and this model just shows that zinc also binds to I think I labeled it wrong, COP-Z. Yeah.
Zinc binds to COP-Z? No, zinc binds to COP-Y.
Zinc?
Under copper deficient conditions, zinc binds to COP-Y, and COP-Z, acting as a chaperone, it brings copper and
to
COP-Y.
Okay.
Did you make this as well?
No, this is from the paper.
Yeah.
Yeah.
They haven't proved actually this, but they have proved that COP-Z can bind to copper, and zinc can bind to COP-Y. But under what they say, when copper is there, then this whole mechanism occurs. But what are the optimal conditions or things like that?
What amounts? Yeah. I'm going to skip that slide. So here's the 3-dimensional structure for COP-A. ATPase in E here.
Yeah.
I think I also have a picture of this one. Let me look if I do. Branch, no. Acetate kinase. No. I want to show you this picture later.
Okay.
Okay, that's a different. That'll be the next hour, I guess.
Okay.
Okay, so the next paper is Rademacher,
Yeah.
copper response gene regulation.
Yeah.
And I think this picture is really confusing to us because here we have copper binding to COM-C.
I don't think the COM-C here is the COM-C we know about.
It's a copper operon.
Yeah. Really? They have COM-C?
In E. coli. Yeah,
Okay.
just recently. I don't know if it's a year or 2 ago.
Oh,
but they published this paper, COM-C, in E. coli. And they have this really nice copper story with COM-C. Yikes. just to throw us off. But in the E. coli, copper binds to QR, which regulates the COP-A gene.
The E. coli system acts very differently than gram-positive system because they have this periplasmic space where they have another set of genes which throw the copper from this periplasm to outside. So they have one system where the copper is thrown out from the cytoplasm to periplasm, and then another system which acts in the periplasm and throw the excess copper out of the periplasm. Now, E. coli requires copper for some of its enzymes, unlike Streptococcus mutans. So they need copper as well, and it's toxic also at higher concentration.
There's a number of transcriptional regulators. So in this table, you've got E. coli transcriptional regulator all the way down to bacillus.
They have COMR also.
So E. coli's got QRQSRS, two-component signal system, and COMR. And here's the Enterococcus herae, again, COP-Y, with the typical helix-turn-helix motif. And as you can see, there's all different kinds of regulation going on by copper. Things can be activated or repressed using copper. So here's again the E. herae picture. Copper goes in to COP-C. COP-C regulates. It transfers the copper to COP-Y. COP-Y is repressing the operon. And copper is also inducing protease of proteolytic functions, degrading the COP-C. Here's a picture showing how copper can regulate post-transcriptional events in capsulatus.
What's capsulatus?
I don't know.
Okay.
I haven't studied capsulatus before, but it has my last name. Almost.
Is it rhizobium?
I think so.
So it's soil bacterium. So in soil they have lots of
everything.
Copper. Okay. So they might be needing copper for their activity as well. Metabolics, I think.
And what they did was they did different kinds of mutations, and they find the structure for the stem is different based on those mutations. In wild type, where you have the regular nucleotides facing one another in the mRNA, then you have without copper, it's not stable, but with copper, this function, the mRNA is very stable. Then if you mutate one or two of those residues without copper, it's very stable, and with or without, it doesn't matter, because the mutation fixes whatever sensitivity it has to copper. Same thing with the second mutation, and in a compensatory mutation, if you mutate it back to wild type, you get the wild type form.
Doing the opposite.
Yeah.
Nucleotides. Wow.
It's flipped.
Still it's working though.
Yeah.
So they just need lock and key theory, right?
Right.
But these are nucleotides, right? Which they are messing with. So if this was like ATGC, I'm thinking about that. So if they have put both A here, so of course they won't base pair with each other and won't perform their function. Here also the same nucleotides won't. They need the complementary
to be functional.
Yeah, to be functional. So they're actually messing up with the structure of the stem.
Right.
So of course they will find this. But they are finding high.
But it's also related to copper.
Copper, yeah.
Yeah, I think
Yeah.
that's the point
Yeah,
of the
yeah.
whole thing, is that it's in the presence of copper,
Yeah.
you find this
Or in the
strange.
absence also they have high.
Yeah.
So actually copper is not doing anything there.
It may confer some stability to the mRNA, and the stability may be needed for function.
But only in the case if that particular structure is there.
Yeah. It might get away with other metals, but in this case they found it with copper.
Yeah.
Okay, here's 2 research papers.
Yeah.
At the end. So the first one is going to be Streptococcus pneumonia.
Yeah.
And the next one is Streptococcus suis. Okay, so 501's free now.
It's okay.
Okay. So if you delete ADCR,
what is
and
ADCR?
Yeah, this is the zinc uptake regulator.
Okay. Metal regulator?
Yes.
Okay.
Involved with zinc. If you delete ADCR and do a microarray, you get the signal for all the bacteria. Then if you do an overexpression and do a microarray, then you get a value for the signal. And what you do is you can then plot the signals for both microarrays against each other. Now where the genes lie off the line, those are regulated, up regulated or down regulated by zinc.
And what about the genes which are in the line?
On the line,
They're
within normal parameters.
So they are not affected?
Yeah.
Okay.
I think this is a pretty good way to show how things are regulated, because then off the line you can go after these genes and actually investigate their functions based on
in real time.
Yeah.
But why not wild type?
I think the overexpression is compared to wild type, and the deletion mutant is compared to wild type. So the wild type is in there, but you lose the wild type information. Okay, the next one is what they did was they grew the mutants to show if there's any sort of phenotype based on deletions. So here are the growth curves of the wild type. In the open circle, that's on top. And if you make deletion mutants, whether it's a deletion of this region ADCR, or if you take ADCR and change the cysteine at 30 to an alanine, histidine at 108 to lysine, or at 111 or 112, you see that there's a definite reduction in its ability. Just a note for the one at 111, 111 doesn't change very much at all. It's right at the top. And this letter X, cysteine 30A, it doesn't change all that much either, but the others do.
Okay.
So that gives you a hint that the
Only
other
these amino acids are essential or performing their duty.
They're performing bigger tasks.
Yeah.
Next, they also determine the amount of zinc that's being in the bacteria. This is kind of like your ICP experiment, and I think that's what they did.
I think they would be done. They have done that only, yeah.
Yeah. So that's one. Now, here's a really complicated way of looking at the enzyme kinetics, is to do these titration curves.
Goodness.
I know. It's quite an elaborate scheme, but what they're showing is they're trying to figure out, so you have all of these mutants, what are the values for the binding constants? And wild type has a binding constant in the presence of zinc of 1400, C30A 710, so that means that C30A doesn't make much of a change. CH42A, so they think that the zinc binds at histidines. Look, it drops down almost a thousand-fold, right? So they know 42 is important, 108 is important, 111 not so important.
Yeah.
It's almost up to wild type levels, and 112 and is not as important. Now, if you check other metals like cobalt or manganese, they don't have nearly as high
Association factor.
rate as wild type.
As zinc.
Sorry, yeah, thank you.
Yeah.
Wild type and zinc.
Yeah.
So these tables or these curves are really complicated.
I don't know why they are using not the wild type with other metal ions, but C30A.
You don't know why. What?
Why they are using C30A strain instead of wild type to see the association of this protein ADCR with other metal ions. They should have used wild type, no?
Oh. I think so. I think you're right. Maybe we'll have to look at the table.
Yeah.
I didn't put it up here.
Yeah.
Do you want to look?
No.
Okay. In this case, they're trying to figure out the numbers for the change at 108 and 112, and it's significant as well. That's the take-home message. And in fact, in these two cases, there's no binding to zinc at all.
Yeah.
Next, they do a spectroscopic experiment, or not a spectroscopic, X-ray experiment. And the reason they do an X-ray experiment is because that if you do this, looking at zinc spectroscopically, it's got a D shell that's filled, so you can't actually look at it spectroscopically because spectroscopic investigations require one extra proton shell. So that's why proton, nitrogen 15, carbon 13, those three work, but in the case of zinc it's even, so you can't do that. So they do instead X-ray, and what they did was they looked at the wild type X-ray at pH 6, and the line there is black, C30A, the line is green, and wild type at pH 8, and there's no difference. It's like,
even
wow.
in the colors of graph, there is no difference. How can one identify what they are doing actually? Anyways.
Well, they're trying to see if this C30A,
Yeah,
cysteine is going to be important with the dimerization.
I don't understand this technique at first place, that if they are saying that X-ray cannot be performed using zinc, then why are they using this technique?
They're using X-rays instead of spectroscopy.
Okay.
So this is an X-ray, and X-rays would work normally. So if there is a difference, you'd see it here.
Okay.
But you can't see it on a NMR
Okay.
as well. There's also this bottom image. So the the X-ray at the bottom shows C30A, and on the inside is wild type. What they're saying is that if you have a zinc bound to histidine, then you're going to have a 3 to 4 angstrom distance. So in wild type, this is the peak at 3 to 4 angstroms right there, and in this mutant, it's gone. So the signal's lost. So yeah, there is binding to the histidine from this zinc.
Okay.
So they did show that it is bound, but they can't show it in this figure, but they do show it in this figure.
Why show those results then?
This is a great little experiment. This is NMR showing the protein structure. On the left you have the absence of zinc, and what you're looking for is a really well-defined peak, and you can see all of these peaks are really well-defined all the way around. And on the cluster, where it's the center of the protein, you see that you have very good resolution of all of the peaks. You can follow those nicely, so it's a good peptide to follow with NMR. This is a first sort of step. Then what they did was they took this information and they lined it up with the presence of zinc. So then they add zinc to it and then they overlay it. And when you overlay the two, you see red being the image on the left and blue being the addition of zinc. Things change. So if you just take a look at that A35, that's moved. L1, this should be L138. L138, that's moved as well, someplace else. Totally different peak.
Mm-hmm.
I93 on the left is very easy to see, and I93 doesn't move, so that means that I93 is not involved in the zinc binding.
Mm-hmm.
So you can figure out which residues
This is a research paper, one research article,
right.
or
So
the
they have
review
the
article?
no, this is a research paper.
Wow,
so they have the
very nice.
microarray,
Yeah.
which is really good. I think
Yeah.
that was a really good microarray kind of thing to do.
Then they have dissociation studies,
Yeah, X-ray,
X-rays,
NMR.
and then this also, where it's total biochemistry
Yeah,
paper.
it's a really good thorough look at the biochemistry of ADCR.
Yeah.
The next one shows, okay, so if you have these histidines, are these histidines important? Now, if you take the histidine, let's say wild type, and just take a look at the spectra, you have this chemical shift spectra. And if you take the histidine at 111 and change that to an alanine, you get the same spectra. So there's no interference by changing that histidine to an alanine. Now, if you take histidine 112 and change that to an alanine, you get no chemical shift at all, the whole region, indicating that all of the signal's lost. And same thing with 108 and histidine 42A. So they're showing that these three histidines are probably the three histidines that are really involved with the binding to zinc. That's the paper. So next paper. Errata. Cation uptake regulators ADCR and FIR are necessary for virulence of Streptococcus suis. So what is Swiss? It's a bacteria that infects pigs. Now if a person is working with a pig that's sick, they could get this bacteria as well. So it's a bad infection. And there's very little understanding of this bacteria because it's not really well studied. And they know that the proteins ADCR and FIR both control the zinc and iron uptake. ADCR is the transcription factor for zinc uptake, and ferric uptake regulator, or FIR, controls iron metabolism. Now if you take mice and you do knockouts of genes, so you delete ADCR, delete FIR, and then do a double knockout. So in the wild type, this is the diamond shape, and you infect your mice, mice with the Streptococcus suis. Notice they all die off.
Yeah.
Now if you take your mice and infect it with the ADCR that has a deletion of the zinc, they survive until day 17. Some of them are dying off. And the other two deletions, they survive. So obviously there's a role in the virulence of this two proteins with what's going on. They show the growth rates for these genes. So in wild type, I don't have the filled triangle there, but the filled triangle there should show you that that's what it looks like growing normally. And this is what I like to see is when the optical density is on a log scale. If it's on a log scale, then differences are really differences.
Yeah.
So in the case of any deletion or a double deletion, all three really affect the growth rate of the Streptococcus suis. What they also did was the survival curves for hydrogen peroxide treated cells. So in the case of wild
Why
type,
they're using hydrogen peroxide only?
they're trying to find a stress that involves metals as well as so they know that fur is involved in peroxide resistance.
I think both iron and manganese, they are involved in producing oxidative stress. So they will use this H2O2 and they will produce superoxide radicals which causes oxidative stress. Now when they are removing these proteins which are involved in their
Do you think it would do better?
No, I think it should be the same case. So
Okay.
they don't have these metalloregulators, right? So
Right.
they don't have these cations also present to produce the oxidative stress. No. Wait. What does these proteins do? They bring in or they take out?
Ferric uptake regulator.
Uptake regulator.
So
So
when
there
iron is
shouldn't be any
in there.
uptake in these cations, in these
It's
mutants.
the regulation based on iron is absent.
Okay.
The regulation based on
Yeah,
zinc
but what
is
are they
absent.
regulating?
Zinc mechanisms, zinc sensitive mechanisms, or iron sensitive mechanisms.
What are those?
That's a good question. That study's not done.
I think it would
Especially
be done.
in Streptococcus.
Oh, okay.
Yeah?
Okay. Yeah.
It might be done in bacillus, but
Yeah.
not for us. So the conclusions from this paper: metallic regulatory genes affect valence, and they also cause bad deleterious changes in the organism.
Mm-hmm.
That's those papers. Did you want to look at anything from those papers?
No.
No? You're good?
I'm good. Thank you. Nice presentation.
You think so?
Yeah.
So that's the there are more pictures in some of them, but didn't think we had to go through all of them.
They're pretty pictures.
No, some of them are pretty. Some of them are really, like this one, color figure.
They have to mention it. It's colored.
It's like somebody wasn't doing their job. Interesting. Yeah, there's another one, another color figure.
Okay. I think it's editor or something. Someone in the.
Somebody forgot to remove the word color.
Yeah.
But this paper was, I think, really strong.
Yeah, it's very. What journal is it?
Journal of Molecular Biology.
Seriously?
JNB, yeah.
Is it good journal?
Very good journal, yeah.
What's the impact factor?
Let's find out.
What happened to my computer?
Journal Molecular Biology Impact Factor. Do you like
3.8.
3.8? I thought it'd be 3.9. It's here.
Yeah, but it's not even JBack level.
I think maybe who's the editor? Editor Elsevier? Maybe because it's not open access, it's low.
Yeah, it's a very nice paper. They should have sent it to a.
I think it's a good journal.
Or maybe
I think it's
they
a good
just
paper.
repeated it in a Streptococcus pneumoniae. It has already been done in
Other bacteria. Could
other
be.
bacteria. So.
Yeah. Okay, we'll stop the program.
Okay.
I think it's Command Shift